Well, thank you for the very kind introduction. I am really honored to be here. I'm honored to be a co-recipient with Michael Burke, a good friend, an inspiration to me, a ball of energy and hope and enthusiasm. I acknowledge also all of the wonderful Colvin Prize winners that have preceded me. I want to acknowledge NARSAD. They funded me as an individual, as a young investigator, then an independent investigator. I've had three wonderful scientists that have worked with me and now have independent careers. They all had young investigators or independent investigator awards. When I was chair of the department, we counted the number of awards that our faculty got, and we were always really, really excited. NARSAD is an agency which takes risks, goes after really bold ideas, and doesn't make you spend a lot of time writing long grant applications either, so they'd rather have you doing the research. So I'm really, really pleased and delighted by that, so thank you. So one little plug for Canada. We have a new Prime Minister, Justin Trudeau. We couldn't have done better, my view. Not as Dean of Medicine, but as a Canadian citizen. But his mother, Margaret Trudeau, as you know, is a huge, huge, huge supporter of mental illness, the whole area. She has bipolar disorder. She's written about it, talked about it openly, and she supported many of our organizations. So I can't see us not doing a lot better in Canada as a result of having Justin in the office for many, many reasons, and that is certainly one of them. So today, let me, before I get into details, let me just put my hat on as a clinician. I don't do very much clinical work right now, but I do, and what I do, I see patients with bipolar disorder. A patient is just now in hospital. He has been well on lithium for 4 years and out of the blue stopped taking his lithium 10 days ago. Maybe he was getting unwell, changed his insight, but nonetheless he stopped and he got incredibly unwell within a week, bought a car, had an affair, disappeared. You know, it was just a tragic nature. And now he's in hospital, he's back on his medication, and a couple of days later, not just lithium, but some other treatments too, and is already getting better. It's a fascinating model for us, and I do acknowledge where Pat was going earlier with our need to be open about diagnoses, but this is one of those sort of models of a really specific biological kind of thing which can just go so badly awry and cause such devastating impact for a patient and their family as well. So to try and understand it, I have been working over the years trying to look inside the cell. This was a sort of new idea when I started with my first Young Investigator Award. And we've looked at different cell organelles, and one of them, which is really interesting, is the mitochondria. Now, mitochondria does many, many things, and I look at Bob Friedman here, and from his center with Steve Dubosky, they were looking at calcium, which is involved with mitochondria. There's apoptosis and involved with cell death, which you've heard from many others. Husseini Manji, very involved with NARSAD, really worked on that area. But then mitochondria is all about energy metabolism, and what could be more obvious about an area of interest in bipolar disorder than energy, where people go through the energetic phase of mania to complete shutdown depressions. So for a variety of reasons, we really focused on this cell, and particularly in the energy metabolism within the mitochondria. So this slide shows a thing, this is all the microarray studies which are done in postmortem brain. So a microarray is a way of doing a non-biased assessment of all the genes that are expressed in a particular illness and use mathematical modeling to pick out which of those are different in different groups. And what was really surprising was in these microarrays, mitochondria came out, energy metabolism just stuck out. And particularly when you look at the blue, oh, whoops, there, got the wrong thing going here. Here we are. The arrow is where was the arrow? The pointer. Do I hit the green there? I'm sorry, pardon me. Blue is down, red is up. What's really interesting in bipolar disorder is there's this real focused, decreased energy metabolism throughout a whole different pathways. Schizophrenia, it's up or down, it's more widespread, but there's something really specific about this in bipolar disorder. So through a series of experiments with a number of colleagues, but most particularly sitting in the front row here, Anna Andreazza, who I'm eternally grateful for having joined my lab and now to join me as a colleague, we just basically went step by step and said, let's just look at each of these proteins, their expression, their function in postmortem brain from patients with bipolar disorder, and from all the different complexes of this electron transport chain, which is the engine of energy within your cell. It makes ATP right in mitochondria, and we have investigated that throughout a number of different pathways. The main findings were really quite remarkable and published about 5 years ago, was that this NDUFS7, which is part of complex 1, was really decreased in bipolar patients compared to controls. And what was really remarkable was the bipolar group is really different than patients with depression or from schizophrenia, and that is, to me, a very interesting finding. It turns out that that was associated with a lot of cellular damage, oxidative damage within this brain region using postmortem brain from patients with bipolar disorder. And this was not as specific, the oxidative damage. That was across many psychiatric illnesses, but it's linked to this complex 1 abnormality, really seemed to be a bipolar story. It was highly correlated and led us to be extremely interested in it. So the first thing we wanted to do is replicate it in another sample. So we had the first tissue was from the Stanley Brain Bank, and the next was from the Harvard Brain Bank, and we basically replicated the study, and we thought we should go a little bit deeper. And so from this postmortem brain tissue, we pulled apart the different components of the cell to see if we could really look at the mitochondria, we could look at lipids, which are involved with white matter, and look at the synapses, which is involved with all the neurotransmitters, which many of our drugs targeted, and looked for oxidative damage within those different areas. And we, of course, used markers with an electron microscopy to make sure we were looking at what we thought we were looking at. And again, we found the same findings there, really quite remarkable. And we found some differences where there were maybe more oxidative damage in synapses, more nitrosive damages in mitochondria, and that's an area that we continue to research at this point. But we also found a really remarkable thing, which was if you look at lipids, which are the things that are part of white matter, which coat all your cells and help neurons transmit and connect with one another, there's a real difference, a real change in lipid damage in the myelin fraction, more damage in the myelin fraction, and we found this both in patients with bipolar disorder and schizophrenia. And it turns out that this is I think a very interesting story because it allows us to look with brain imaging and in the periphery, and that'll be the segue into the next little part of this presentation. So doing postmortem brain studies are very important, and we are incredibly grateful to the generosity of patients and their family that allow us to do that. But it's not a practical thing that we can sort of spin out into a diagnostic test. So the best thing, of course, is to have a blood test if you can, something where you can take a tube of blood and you can measure that very easily. And Michael talked about biomarkers. And so that was kind of, we wondered whether this oxidative damage could be found in blood cells and whether those blood cell measures might somehow relate to the central abnormalities which I just talked to you about. And let me go through some evidence which supports that. So again, Michael told you all about meta-analysis, so I don't need to do that today. But when you do that, that's a way of looking at all the literature that you can find. You do qualitative studies. You throw out some stuff that you don't think is very high quality. You take the stuff which is high quality, you put it together, and you can then get scale up your findings, and you can see whether it's something which is perhaps more meaningful. And as it turns out, when you look at different markers, a couple of these things stand the test of time in meta-analyses, and one of them was lipid peroxidation, and another of them was DNA damage as a result of oxidative stress. So here's the forest plot which shows it, and it's a pretty robust effect, particularly when you look at the lipid peroxidation, and you really do see a lot of evidence of this in patients with bipolar disorder. And let me just show you a couple of examples of them. We also had an opportunity with some colleagues, Benoit Molsant, who's now our chair of psychiatry, to look in blood samples of a group of geriatric age patients, only 65, which I don't really think is sort of, as I look ahead, that doesn't really sound very geriatric to me. Maybe, I don't know, 80 maybe is at the beginning. I don't know. It's 80 is the new 50. What is it they say? Or 65 is the new 30. So that's how I'm seeing. That's my plan for aging, is to see it that way. But we were able to look at the sample, and they were very well characterized, and they were well, and we had a large sample. And what we found was this peak where you actually could find lipid peroxidation in very well patients that had a lifetime of illness. And our interpretation of it was, not only was this interesting, but it also suggested an active biological process which was there, which predicted the need to be ever present and ever mindful of the fact that you need treatment, you need to stay well, and it was something which you could identify in that sample. So that was the first thing we learned about that one marker. The next thing we did is with Anna Andriaza and a couple of colleagues, Amelia Versace and Mary Phillips at University of Pittsburgh, we were able to collaborate where they had wonderful MRIs looking at white matter tracts and blood samples, and we were able to do a lot of different measures of the different oxidative damages there. And we found this really remarkable correlation between the amount of white matter damage you had and the amount of lipid peroxidation that you had, and I thought that was very interesting. It was like a blood sample measuring changes in brain imaging, which could be relevant for you having bipolar disorder. This was found across a number of patients, but it was particularly relevant in patients with bipolar disorder. Now, this work is continuing and underway, but you can see here, these are two measures of white matter changes, and you can see they're different in bipolar patients, and you can see this measure of lipid damage, peroxidation, is different in bipolar groups, and I just showed you the correlation. There's some similarity in schizophrenia, but again, it's different. Certainly there are white matter differences there, but they look different, and this link to the damage to lipids through peroxidation is really not a marked finding in patients with schizophrenia, suggesting some potential specificity for this. So then moving to the DNA damage, which also could be measured in blood samples with our colleagues at University of São Paulo in Brazil, we were able to collect a number of patients where we had very clear evidence of how many episodes of mania they had, and we're able to imagine that maybe there was a cumulative effect of mania, so that the more episodes you had, the more damage you had. And we were able to show in this study, first of all, that DNA damage as a result of oxidative stress in blood samples was predictable. And then we also were able to show this very close correlation in a relatively large number of subjects, not 19,000 like Michael has. We'd like to certainly do that in 19,000 subjects, but a relationship between the number of episodes and the damage was there, suggesting an active process which could be underway and which is something that we might be able to target by some of the treatments that Michael has mentioned in his talk. So another important aspect of this is trying to understand our innate ability to deal with any oxidative damage, and glutathione, as we know, is our innate antioxidant, and NAC is a synthetic glutathione. It's many other things, but that was the main reason you do it, is to bump up your innate antioxidant ability. And this is a study from colleagues at Oxford, where they were able to look at patients that were generally healthy with bipolar disorder with just a few symptoms, and were able to show that despite that, they still had some evidence of potential for oxidative damage, and they had lower levels of plasma glutathione, again suggesting that the system may be something which is really particular and specific and important to bipolar patients, and again something that could be active, could be something that could lead to many more problems, and underscores the need for people to stay on treatment. So like my patient I described earlier, needing to stay on treatment, how an active process like that, things could go wrong very quickly when you get out of sync with the treatment that you need. So in the final few minutes, let me link back. I think it was Michael's, he said that inflammation is the new Jerusalem. Well, interesting comment. I think inflammation is, I think if I asked, if students come to me and say, what should I study now? I'd say inflammation or microvascular illness. I think those two things are really important for the field, and let me make a couple of comments about those as I close. So inflammation is linked with our mitochondrial function, and there's this thing called the NLRP3 inflammasome. And basically what happens is when mitochondria gets activated, it can link up and it can lead to production of a number of different interleukins and things in the system, which can lead to cell damage and cell death. And so that's a potential downstream target of these, of changes which are there, and one that many of us are exploring, as Michael talked about, another possibility for treatment. So maybe you fix up the mitochondria with ubiquinone, you fix up the person's ability to have antioxidants by giving them NAC or glutathione, and then you give them some anti-inflammatories to cut down the inflammatories. So a way of stepwise going about it. And it's exactly what we do in heart disease and hypertension and any of those other illnesses. We have a multimodal targeted approach. And this model suggests this could be a biological model to support some of that work. So again, using postmortem brain, similar sample, and this is a detailed slide, which maybe you don't need to go into now, but it just says you have to do work carefully, or if you want to get good results. And what we were able to find was, again, in this sample, oxidative stress and activation of this inflammasome, first of all, looking particularly in the mitochondria. If you look at whole cells, you don't see anything. If you pull out the mitochondria, look carefully, you see it. And then when you look carefully in mitochondria, you see the inflammasome activated, the ASC caspase as well, and then a very interesting pattern, which we're just in the midst of studying more fully, looking at some of these inflammatory measures which are linked up with activation of the inflammasome. So all of this has hope for us developing biomarkers, and I think many of us would like to know that. We would like to know for ourselves and for our family member, is there something that I can know that helps me go to get a personalized treatment? Is there something I can measure in my blood stream, for instance, or with a brain scan that tells me I'm on the right treatment, I'm going to stay well? Is there something that we could see that goes off before patients get unwell, like my patient earlier? Is there something that we could have detected earlier? And there's a lot of interest in our field in that area. Michael Burke, Anna Andreazza, many other colleagues have been part of a number of different task forces, and this is one from the International Society of Bipolar Disorders, suggesting a number of different biomarkers, and oxidative stress markers, BDNF, anti-inflammatory markers are all within that field. So a couple of closing comments. I think I've said a couple of closing comments about 8 times already. So, and I also realize that I'm between you and lunch, you know. So I better wrap it up quickly. Oxidative stress, great pathway for understanding lots of illnesses, and this is an article suggesting how this may help us understand cardiovascular risk. Cardiovascular risk turns out to be incredibly interesting for bipolar disorder. A really bright colleague at our place, Ben Goldstein, just published a great article. You may have seen it in the New York Times. It's been everywhere, suggesting if you have a mood disorder when you're an adolescent, you're more likely to have early cardiovascular risk. Could it be that this is one of those pathways which is getting turned on? Could it be that where we are is in psychiatric illnesses is really trying to just follow the pathway that many of other colleagues have done, which is understanding inflammation, understanding microvascular risk. There are many other parts to our field, but those are sort of core pathways which I think we're really supporting and really revitalizes our field as we go forward. So again, biomarkers, I mentioned some of these priority questions, and this is from a Lancet article, a very good one by Mary Phillips and David Cutfer. and a way of thematically looking at it and bring it more into a wholesome view that Pat McGorry talked about a little bit earlier, talking about different platforms in the illness. And then, luckily, they picked out one molecular dimension. They happened to pick out oxidative stress. Now, we were collaborating with Mary at the same time she was writing that paper, so I don't know whether she was more favorably predisposed to oxidative markers at that point, but it just suggests that maybe symptoms, as well as different markers, genetic predisposition, putting them together may help us have a much more fulsome and rich understanding of where we should go. I have been incredibly fortunate and blessed in many ways throughout my personal life and also in my work life. I have had such wonderful support from NARSAD, from federal government in Canada, many, many opportunities. I've worked with great patients, many wonderful clinicians. I've had wonderful collaborators, Michael Burke, Anna Andriaza sitting here in the front row, among many others. I'm just delighted to be here, and thank you so much for your attention.
Trevor, you talked quite a bit about oxidative stress and bipolar disorder. Could you define that for the group of
Sure. Yeah, so oxidative stress is, so when you burn off energy, it's a good thing, but the byproduct of is that you could produce free radicals. In all things being considered, you clear all those free radicals, having antioxidants in your system, and everything works well. If something goes wrong in the oxidative metabolism, you get damage. And you can clear it for a while, and then it builds up, and when it builds up, it can damage any tissue in your body, and we think that that's a fundamental abnormality that could happen in bipolar disorder, many other, Parkinson's disease, we think that's important, dementia as well, schizophrenia as well, in addition to a number of illnesses of other organs than the brain.
Yes, ma'am.
My son actually has been taking LAC for many years now, and I talk loudly. I may not need it.
I do too, so I get it. So.
No, my son has been actually been taking NAC for many years now and has an extremely positive effect from it that he's noted himself. And this may be a silly question, but I'm going to throw it out there anyway. They've always cited that tumeric is a very natural anti-inflammatory. Is that a different kind of inflammation?
Yeah. Natural products, great, great story. I don't know the specific action of turmeric, as you mentioned. I think it's a pretty broad spectrum anti-inflammatory. I don't know which pathway it is and whether it would affect these. So on the question that you raised, so Andy Nuremberg, who's one of the smartest guys around in any field, we had a nice conversation at a meeting at ISBD, and we put together this marginal gains paper, and it's just out in the last couple of weeks ago. And it was after that model of what athletes do, which is you add on all these different things. So it was like, okay, you take lithium, it works, but then you got to moderate your sleep. You should eat well. Look at supplements which are in there. You should exercise, do some mindfulness, do some CBT, get a good social network. Nothing alone is going to give you that effect. Maybe the medicine has a 50, 60% effect, but if you add each of those, you start scaling it up, another 2%, another 4%, another 6%. So, things like a little bit of NAC, a little bit of, anti-inflammatory, turmeric. I mean, I'm not telling you, I'm not saying as a physician, take turmeric. I don't know that, but eating healthy and being aware of what could be in your diet, which could be having positive effects on inflammation, on stress, all those things are incredibly important. That's how we treat every other illness. You see your cardiologist, they don't just, give you a stent, but they also say, are you exercising? Did you quit smoking? Are you doing it? And So mental illness and mental health, that whole area, and our field has to step up to the plate of being able to deliver those fulsome solutions for our patients. And you know, I'm looking at Pat here in the front row. That's the headspace thing. You get it every way, and you put it all together, and that's that kind of outcome you're going to get. So I see a theme throughout all the talks this morning, which were all about kind of us taking ourselves out of our comfort zone, you know, in neurotransmitters or dopamine, and getting out to the edges and thinking about all the different things we can bring to bear positively in our patients. And I think that's the hope for our field going forward. Do you want to grab the mic?
Hi. Yeah, my question is about biomarkers. You talked a lot about them in your presentation, and I know that some companies are already marketing these genetic assays and suggesting that you can use it to pick your drug. Do you feel that those are clinically ready, or are those really too soon? What can be really done with those?
Yeah, so I think a great question. I don't think there's any clinically ready biomarker at this point. That would be the overall question. I think some of the genetic markers. I see my colleague here, you know, Jim Kennedy, another Torontonian who's also, and his partner, Kathy Barr, wonderful friends and wonderful colleagues. And, you know, Jim is doing some great work looking at genetic variability and the enzymes that metabolize drugs to predict which antidepressant you come on. I don't think we say it's clinically ready yet, but it looks very, very promising. Or somewhat promising, so I think it's a ways before we can do that. You know, but I think we have to collect more data, is my view. I don't think we got to elevated cholesterol and the lipid profiles being so important for cardiac disease without collecting it on a lot of different patients. And to go to the idea of stigma and how much we fund is, it doesn't take a lot to ask to get a trial to add 3 blood tests to a cardiology trial or get a new $20 million some kind of machine to measure patients. You add that to the trial. We ask that in mental illness, and you know, you're scratching your head. Do we really want to fund it? So I think we have to be generous. We have to be somewhat expansive. I don't see a lot of danger in us collecting an extra tube of blood and looking for more things and collecting that data, even though it's not clinically relevant. Maybe when we do it and we get it on a large enough scale, we'll be able to answer some of those questions. Everybody wants this knowledge. People are out there doing it. 23andMe and all those questions. People are doing that all the time. They want to know what's in them that's going to be important for illness.
Okay. Yeah, one last question over here.
You are all researchers, and you're doing as good a job as you possibly can. But you mentioned about stepping up to the plate in regard to the comparison between those that try harder to find results in other illnesses. Well, the fact is, how do we get around the advocates and all? I know the brain, as you know, is the most complicated organ in the body, and I think that is part of the reason why there's always so much hesitation in trying to get to some new things because of that. And that shouldn't be. That's part of the reason. It took so long to even get where we are now because of too complicated. Many factors in the brain that control this, that, and the other thing. But don't you think that we ought to, as a group of advocates too, try to get this thing across where we have to step up to the plate more?
So I couldn't agree more. There's lots of reasons to be hopeful. Yes, dean of the medical school now. I don't spend a lot of time in psychiatry, but I see the brightest students going into psychiatry. It wasn't like that 25 years ago. I see, our place hospitals getting donations in the area of mental illness. The changing. I just told you about our prime minister and his mother with mental illness. I think the field it's just ready for us. It's wearing down. Battling mental illness for patients and families, it's really wearing because it starts young and it stays with you a long time. But there's every reason in the world to be hopeful about more interest and more. The best is yet to come, is how I would see it. So
thank you. Thank you, Trevor.